A very good evening. A very good evening to all of you who have gathered here for the first Dr. Suniti Solomon Memorial Lecture. We would like to extend our warm welcome to Mr. Kannan, Professor Ravi, Dr. Krishnamurthy, Ms. Corey Bickel. A special welcome to our advisors, Dr. Ramanan, Mr. Ramakrishnan, Dr. Jacob Kumaresan, Mrs. and Mr. Balram, who could join us this evening. We would like to welcome Mr. C.P. Rao, Mrs. and Mr. Kamdar, Dr. Srinivasan, Mrs. and Mr. Shivakumar, Ms. Ratna Mukherjee. A heartfelt welcome to Dr. Sunil Solomon, Mr. Krishnan, Mr. Ganesh, colleagues and friends from YRG Care. I would like to welcome Mrs. Kamla Ramamurthy and Mrs. Janaki Kannan, who are leaders of our Chetna and Alaigal programs of Setu Foundation, along with Ms. Jessie, who has immensely contributed to our Chetna outreach program. I would like to welcome Mr. Parthasarathi, our General Manager of Finance, who has taken time off to be with us this evening. A big welcome to all students, friends, and admirers of Dr. Suniti Solomon, who have made the time to log in today from India and from across the world. I would like to welcome my friends and well-wishers who have honored our invitation this evening and have made it here. I am privileged to announce that we cannot have a better person than Professor Ravi for the memorial lecture, who has also been Madam's student during his undergraduate days and an acclaimed virologist today. We're equally privileged to have Dr. Krishnamurthy introduce Dr. Ravi with us. Each of them are renowned medical professionals in their own right. I should take this opportunity to introduce Setu Foundation as a non-profit company registered under Companies Act of 2013. Our abiding theme is unity of life. We presently work for Chetna, meaning consciousness, programs for children, and Alaigal, meaning waves, programs for women. I would like to welcome our collaborators and donors this evening, United Way of Chennai, Nidhi Gupta of Roundtable, Sophia Kandelwal of Roundtable, Devi Krishna of Rotary Club, Himanshu Budhia of Roundtable, Kabir Singh of Roundtable Mylapore, Ginny Singh also from the same club, Jasneet Kaur, Sunanda, Shivakumar, and Ranjini. Many of you may know Dr. Suniti Solomon in the world of science and a pioneer in the field of HIV. But I have had the privilege of knowing a very warm, exceptionally humane person. She always used to say, scientific pursuit in YRG should help the poor and the needy. A wonderful person whom we used to call Madam most affectionately. We are both humbled and fortunate to host the first memorial lecture to honor her legacy. Thank you all. We will now have Mr. Sivan Kannan, IRS, who's the Chief Commissioner of Northeast Zone, headquartered at Guwahati. He's the person behind the idea of this memorial lecture, and therefore, I would like to invite him to share the story behind it. Sir?
You know, Saint Tyagaraya could not have put it better. In terms of salutation, he said, Endaro Mahanubhavalu, Andariki Vandanamulu. I can't present salutation to each one of you, but he says, all the great people who have assembled here, to each one of you, my humble salutations. My talking in this program is very, very unlikely because I never knew Dr. Suniti Solomon. I'm not from the medical field. I don't speak in such seminars where such eminent people speak, but somewhere I got connected with it. It all happened way back when the world was not having pandemic. That is in February. I was traveling back with Setu Lakshmi from Madurai, and I was listening to her work experience and life. And she said she has worked with Dr. Suniti Solomon for about 22 years. And she kept on talking about Dr. Suniti Solomon, and something rang in my head, a bell, that this name I have heard somewhere. You know, then it struck me that way back in 1987, when we were writing the civil service exam, we used to have a 2-mark questions for writing about the great personalities of that year, if they are figured in literature, culture, sports, science, medicine, and all. So we used to sort of mug up the names and what they have done in 1 or 2 sentences. So in 87, Dr. Suniti Solomon's name we had remembered for our exam purposes, and then merely forgotten it. I haven't cleared that exam. And after 30 odd years, here is a lady who is telling me about Dr. Suniti Solomon. Then I said, then I took more keen interest to listen to her story and about this discovery of HIV virus in India and isolating and things like that. Then I said, why don't you, if you have got such a rich experience and association with her, why don't you start some program for her memory? And to begin with, you can have a memorial lecture. The idea got seeded somewhere in the halfway from Madurai. And by the time we came to Chennai, it got incubated into a fairly good idea. And the question was, whom should we invite? I knew Dr. Krishnamurthy because he is one of the leading neurologists, and he is in the next street to ours in terms of office location. I told her, why don't you go and request him, and he will guide you. And Sethu Lakshmi went, and Dr. Krishnamurthy, I think in minutes solved her problem and directed her to Dr. Ravi, and the rest is, as you say, it is history. Dr. Ravi accepted, and here we are to listen to Dr. Suniti Solomon first memorial lecture. That is how I got connected with this whole thing. Very, very strange association that I am sitting in Guwahati and I am able to connect with all of you eminent people. Thanks for this opportunity to talk before all these big people. I'm sure there could be nobody better than Dr. Ravi to deliver this first memorial lecture, which will become a tradition year after year, and which will honor the memory and legacy of Dr. Suniti Solomon. And what she did for India in terms of isolating AIDS virus or the HIV is very interesting, because we were always in denial mode, I think till 1986, that India cannot be affected by this virus because culturally we cannot attract this virus at all. I mean, we are sort of morally and culturally above this virus. The virus also had other non-medical dimensions which were very interesting, and it is still interesting even today. But somehow we came to know that we are as much human as all others in this planet, and we are fairly attracted by this, or the virus attracts us, and we are infected, and things happen. So this is one great story about Dr. Suniti Solomon, which she recounted and which we could transform into a memorial lecture. And today, on her birth date, it's very, very gratifying to see so many people from all over the world joining together to pay tributes to this great lady. Thank you very much, Sethu, for inviting me and allowing me to tell our small story behind this memorial lecture. Thank you very much.
Thank you, sir. Thank you very much for your kind words and for that lovely message about how this entire thing started. We will now have Corey Bickel, who will offer the opening remarks. She is the press officer at the US Consulate General in Chennai. She previously served as press officer and cultural affairs in the Kyrgyz Republic. Her previous foreign services posts include Hermosillo, Mexico, Vientiane, and Laos. Corey is a graduate of the University of Maryland and Emory University, where she did her MPH. Prior to joining the Foreign Service, Corey was a program manager at the US Center for Disease Control and Prevention of Central Asia Office in Uzbekistan. She was a Peace Corps volunteer in Uzbekistan between 98 and 2000. Corey is an Atlanta native. Very, very happy to have Corey amidst us. And I would like to request her to offer her opening remarks.
So good evening. As Setu said, I'm the press officer. My name is Corey Bickle at the U.S. Consulate General here in Chennai. I arrived only last month in this wonderful city. I'm slowly trying to discover as much as I can about Chennai and Tamil Nadu and South India, though of course mostly from the comfort of my home for the time being. First, I want to thank the Setu Foundation, particularly Ms. Setu Lakshmi, the founder of the Setu Foundation, and Mr. Kanan, advisor for the Setu Foundation. Thank you for those very eloquent words. Actually, it was very, very striking to hear your sort of history of your experience with Dr. Solomon as well. So thank you for inviting me to make the opening remarks for this first Dr. Suniti Solomon Memorial Lecture. I was truly humbled to have been provided the opportunity to participate in this virtual program to honor one of India's greatest warriors against the HIV/AIDS pandemic. I also want to acknowledge our distinguished speakers, Dr. Krishnamurthy and Dr. Ravi. Given the situation that we all find ourselves in today, I was of course very struck by the title of today's memorial lecture, Virus, the Eternal Reality. Communicable diseases and epidemics and pandemics are not new to mankind. As we look back 200 years, the world has witnessed several global pandemics: the Spanish flu, the Asian flu, HIV/AIDS, H1N1, swine flu, Zika, SARS, and of course the coronavirus that we are all being impacted by now in so many different ways this year. We're all also witness to the devastating impact of these pandemics, and especially HIV/AIDS. According to various reports, more than 76 million people have been infected with the HIV virus. About 33 million have died because of it since it was first detected in 1981. People are often surprised to learn that today there are nearly 38 million people living with HIV in the world, including in India. Which brings me to the incredible life and work of Dr. Suniti Solomon. When I was invited to make the opening remarks this evening to honor such an extraordinary human being, I became very inspired by Dr. Solomon's story and her curiosity that led her to detect the first ever cases in India in 1986. And what I learned was that Dr. Solomon was not only a scientist, a microbiologist, and a professor at the prestigious Madras Medical College, but also a great humanitarian. Dr. Solomon knew that her fight was not just against a communicable disease. She understood that the social stigma that was associated with HIV would be the most difficult hurdle to overcome in her efforts to fight the HIV pandemic. And fight she did. By starting the YRG Center for AIDS Research and Education in Chennai, which I understand is currently operating in over 16 cities across India. I have no doubts that Dr. Solomon's efforts and that of YRG Care have helped prevent hundreds of thousands of deaths in India by not only providing clinical care, but educating millions on how to deal and safeguard themselves against the HIV virus. And most importantly, trying to remove the social stigma around the pandemic by coming forward to get tested and treated. And Dr. Solomon's efforts were recognized not only by the people in the governments of Tamil Nadu and India, but also by the United States. The U.S. Agency for International Development, USAID, and the U.S. Centers for Disease Control and Prevention through the U.S. President's Emergency Plan for AIDS Relief, which PEPFAR, not only supported Dr. Suniti Solomon and YRG Care, but has also worked very closely with the government of India and the government of Tamil Nadu in the global fight against HIV/AIDS for over 2 decades. And PEPFAR continues to be active in 6 states across India, focusing on prevention and testing. treatment and care, retention and viral load suppression, and also programs for children living with or at risk of HIV. And YRG Care continues to be an important partner with USAID, CDC, and PEPFAR. After Dr. Suniti Solomon identified India's first HIV case in Tamil Nadu, the government of India set up the National AIDS Control Organization in 1992. And since 1995, USAID has worked in partnership with NACO and other organizations to achieve the current longer-term government of India's vision towards reaching HIV/AIDS epidemic control in the country. And of course, Dr. Solomon can be credited as one of the key people who made this bilateral partnership happen. I have no doubts that if Dr. Solomon were with us today, she would have been at the forefront in the global fight against the coronavirus. Dr. Suniti Solomon was an embodiment of grit, determination, hard work, and the belief that she could make a change that is continuing to create awareness and save countless lives across India. Her work and her legacy will never be forgotten in India and the United States. And as I learned more and more about Dr. Solomon, I also shared her story with my children, explaining to them what a hero that she is for her efforts to make the world a better place. On a personal note, I come from Atlanta. Like Setu mentioned, Atlanta, as some of you may know, is sort of a public health capital of the United States. We have Emory University, the Centers for Disease Control is there, the Carter Center. So as a child growing up, and when I was studying my master's in Atlanta, public health was something that we were always, as children, very very aware of and very attuned into the sort of heroes that were working at some of these organizations. I would like to thank Setu Foundation once again for giving me the opportunity to speak and pay my tribute to an extraordinary woman. And I commend the foundation for hosting today's memorial lecture, which I believe is also the birth date of Dr. Suniti Solomon. So finally, thank you to the YRG Care Foundation for your work to carry forward Dr. Solomon's legacy. Thank you.
Thank you, Corey. We would now like to move on to the main part of the event, where we will now request Dr. Krishnamurthy to introduce our main speaker for the evening. Dr. Krishnamurthy is the president of the International Neuropsychiatric Association and founder CEO of Buddhi Clinic. He is an international academic leader with multiple professional affiliations in the fields of neuropsychiatry, epilepsy, dementia, in the fields, and then published over 70 plus peer-reviewed publications and 30 plus book chapters. He has edited the Global Approach series of books for Cambridge University. Dr. E.S. Krishnamurthy is a recipient of the President's Medal of the Royal College of Psychiatrists, UK, and elected fellows of all 3 Royal College of Physicians based in the UK. Dr. Krishnamurthy, may I now request you to introduce our main speaker.
Thank you, Dr. Sethi, and good evening, everyone. Professor Ravi, Mr. Kannan, Ms. Corey, it's an honor to be here today. And as they say, the universe has a wonderful way of connecting the dots. And I'll explain that a little bit further. I knew of Professor Suniti Solomon as a very eminent teacher in Madras Medical College, where my father was a professor of neurology. So I knew of her through him. And of course, I think we were all made aware of her efforts to HIV. I also had the opportunity of meeting her in my capacity as Honorary Secretary of VHS, the Voluntary Health Services, where YRG Care was based, on a few occasions, and was also always struck by how sharp and on the ball she was and how well prepared she was for her meetings. Apart from the YRG Care work, the diagnostic and clinical care work, one of the other things that happened in VHS, and I think that Madam had a role to play, at least in the beginning, was the entire social impact effort when it came to HIV. And a lot of that was the AIDS Prevention and Control Program supported by USAID, which was managed by VHS for 13 long years, possibly even more. And that program was a health behavior change model program. And I think it worked wonders. And it worked with communities, it worked with people, it worked with those affected, and it was wonderfully conceptualized. And I think so VHS, the Voluntary Health Services, was extremely fortunate to have both the clinical care diagnostic wings, research wing of biology care there, and the APAC project, making a community and social impact. So I do know of Madam in all these capacities, and her reputation obviously precedes her, and it's therefore a great honor to be here today to introduce Dr. Ravi. I first knew of Dr. Ravi when my father introduced him to me. He was our visiting professor for the 1993 T.S. Srinivasan Endowment Oration, which incidentally has happened every year for 40 years. And it even happened this year in February 2020. And Professor Ravi was a young neurovirologist in NIMHANS at that time. And of course, I've stayed in touch with him over the years. He has been extremely helpful to practicing doctors like myself in Chennai by opening his lab to doing tests on difficult patients where we are suspecting virus infections of the nervous system. And his lab is not only very efficient in telling us how to send the sample, but also giving us reports and communicating those reports to us to help us in the clinical care of our patients. So, that has been one way of regular connect. And more recently, a few years ago, when he was the registrar at NIMHANS, the T.S. Srinivasan chair, which was in VHS, actually moved to NIMHANS. And Dr. Ravi was responsible for that establishment as the registrar, together with the then Vice Chancellor, Dr. Satish Chandra. So many dots to connect, and I'm so happy to be here today to introduce him. He's at present the Senior Professor and Head Department of Neurovirology at NIMHANS. He is a student of Madras Medical College and hence of Professor Suniti Solomon. He got his MD Microbiology from JIPMER Pondicherry in 1983, and he established the Department of Neurovirology in NIMHANS in 1985 as its founder. And he has been the head on more than one occasion by rotation, once until March 2016 from the time of founding, and again since April 2019. And his main interest is public health virology, the pathogenesis and immune response of viral infections of the CNS, and development of indigenous diagnostic assays. And he's been responsible for work in the field, investigation of several viral diseases of importance, such as encephalitis, dengue, chikungunya, H1N1, and now COVID-19, for which he is the nodal officer for the diagnostic laboratories in Karnataka state established by ICMR. His research has centered on the development of rapid, sensitive, and specific diagnostic methods for the diagnosis and immunology of virus infections, and he has developed 3 important indigenous diagnostic products together with Excitone Diagnostics in Bangalore, ELISA kits for the detection of antibodies in HIV, Japanese encephalitis, and cysticercus. In addition, he has developed HIV-1 subtype C specific real-time PCR viral load assay, and which is awaiting technology transfer to the industry. And indeed, this is a strain of the virus that many of us, including neurologists, are very interested in because the manifestations are quite different. And NIMHANS has done path-breaking work in describing those differences. He has also developed a macroarray chip with Excitone Diagnostic Industries that is capable of detecting 16 viruses that cause acute encephalitis syndrome. He also has done seminal work in the field of psychoneuroimmunology, both schizophrenia and obsessive compulsive disorder, together with his colleagues in NIMHANS, and currently is engaged in a large CDC-funded project on strengthening surveillance and diagnosis of acute encephalitis syndrome in India. He is the recipient of numerous research grants from both national and international bodies, including the ICMR, the DBT, the DST, CSIR in India, the NIH, CDC, USA, and MRC from the UK have all supported him. He has a European patent to his credit. He has over 170 research papers in peer-reviewed journals and 22 chapters in books and monographs. He's a master trainer for NACO, and he works with several technical research groups, and he's been on various scientific advisory committees of many national institutes in India, and works on national task forces of the ICMR, the Department of Biotechnology, and is an NABL assessor for many years. He is also a teacher who has mentored 25 PhD students and has guided several MD psychiatry students and DM neurology students through their dissertations. He has served as a PhD examiner in all the major institutions of the country, and for MD virology in the Medical Research Institute in Colombo, Sri Lanka. He has received several important awards, including the prestigious Sir C.V. Raman Young Scientist Award, the Dr. J.B. Srivatsa Oration of the ICMR for the eminent medical virologist, the Dr. S.C. Agarwal Award, and the Dr. H.I. Jala Award of the Indian Association of Medical Microbiologists, and the Eminence in Virology Award of the Indian Virology Society. He has been both a Commonwealth Fellow and a WHO Fellow, has been decorated with the fellowships of the National Academy of Medical Sciences, the Indian Academy of Medical Sciences, and is a distinguished international fellow of the American Society of Tropical Medicine and Hygiene. He has worked with the WHO on a number of projects, and he continues to serve as an honorary visiting professor of microbiology and immunology at the University of Liverpool since 2013, and has been a secretary of the Indian Association of Medical Microbiologists 2004 to 7 and president in 2011. I know him as a very efficient administrator and as a wonderful human being, in addition to all these accomplishments. And his hobbies include cooking, conducting quizzes, and writing rhymes. So I'm sure that we are in for what I'm sure is not just scholarly, but very, very interesting lecture on viruses, coming at a time as it does when we are all amidst a pandemic caused by a virus. So thank you, Dr. Setulakshmi, for the opportunity, and I put all of you back to her for her moderation from here. Thank you.
Thank you, Dr. Krishnamurthy, for that lovely introduction. May I now request Professor Ravi to take on this floor to the much awaited audience.
Good evening and good morning and good afternoon to all participants wherever you are. At the outset, I must say it's a great honor and a privilege to give the first oration of a beloved teacher. There can be nothing more higher recognition than this for a student. I would like to thank Dr. Setulakshmi and the foundation and YRG Care. I would also like to thank Sri Kannan, Ms. Kori Beckel for their opening remarks, and my dear friend Dr. Krishnamurthy for his personal touch that he gave to the kind introduction. Krish, I wish whatever you said was true. Thank you all very much. So I will, what I have been given this title by the Setulakshmi Foundation. I found it very apt, and so I decided not to change it. Once I decided on the title of the oration, then it set me thinking on what am I supposed to talk. Before I do that, let me pay my respects to my teacher, Professor Suniti Solomon. I love this picture of hers, which I've chosen from the net in white and black. She was such an honest, warm person, outspoken, a fantastic teacher. As an undergraduate, we measure teachers in undergraduate theory classes by not sleeping, and I could not sleep in Dr. Suniti Solomon's classes. I knew her as a very bold person, very courageous. What struck me most was the passion with which she worked in the field of HIV. She's bold and courageous because she gave up a wonderful academic career to tread on a path that's new, and she created her own path and left behind her footprints. She was compassionate and caring. Most of you know that from the kind of work she did with HIV, especially women and children. And above all, she was extraordinarily committed to her work. So, happy birthday, madam. I'm sure you will be in heaven. And that picture was a picture I got from the net where probably she was doing the first ELISA when she detected the first case of HIV in India. I'll start by telling you what has influenced me apart from my teachers, great teachers. Books have always fascinated me, and there are 4 books that I recommend that people read. The first and foremost is a book called Dancing Matrix, published in 1993. An extraordinarily great book to read because it predicted what is going to happen in 1993, much before we faced many of these pandemics. The second is Viral Storm by Nathan Wolfe, which tells you what we should be doing for the future. The third is a very philosophical book, one of my favorites, The Alchemist by Paulo Coelho. And whenever I read the statement, the main message in this book, that when you want something, and if it's for the good of others, I believe, all the universe conspires in helping you to achieve it. And Professor Suniti Solomon is an example of that. And the last is a very fond book of my classmate, a great physician, a great crusader on HIV/AIDS in the United States, Abraham Verghese. I call him Abe. These books have left a very profound impact on my thinking and my career. So those of your book lovers, I encourage that this should be in your collection. What I will do in the next 35 minutes is to go through the lecture. I will start with trying to understand the literal meaning. That's the first thing I did. I opened dictionaries and looked for what is a virus. A virus is a kind of a germ that can cause disease. What is eternal? Anything that lasts forever and doesn't have an end is eternal. And what is reality? The state of things as they actually exist. In common language, they are real bugs and they're here to stay. That's the message I got from the title. So I thought I will try to address this in the form of questions. Where do we stand today as far as viruses being eternal reality? What are viruses? Why are they here to stay forever? Why do they exist? How do they periodically emerge and how do we tackle them? How do we tackle the future emergence of viruses? I think something which Nathan addressed in his book. What are the challenges and opportunities, especially in the current context of what we are all going through, difficult times of COVID. So what I will start with, where do we stand? Where do we stand in terms of viral infections? If you look at the past century, there have been a number of viral outbreaks. You can call them outbreaks or pandemics. Some of them have been huge pandemics. A century back, we had the Spanish flu. that claimed 50 million lives. And since then, periodically, we have had various outbreaks of flu, the Hong Kong flu, the Asian flu, the Russian flu. And then, of course, we had the emergence of Ebola in 1976. And down to the late part of last century and to the present century, you can see that there are a number of viruses that have been systematically emerging in the last several years, especially in the last 2 decades. What are viruses? Viruses are ultramicroscopic organisms. You cannot see them under the light microscope. They consist of only one type of genetic material. We all know genetic material can be RNA or DNA. And they're very simple in their structure. They're surrounded by a protein coat. Some of them may have an envelope and others do not have envelope. By nature, they cannot survive on their own. They need another living system to survive. Sir Peter Medawar summarized it beautifully, saying that a virus is a piece of bad news wrapped in a protein coat. So these are viruses. Why are they here to stay forever? There are 4 inherent advantages viruses have. The first and foremost being their ability to replicate rapidly. Many viruses can produce millions of particles in 24 hours. Take the example of HIV. In a matter of 24 hours, it can produce up to 1 billion particles. The second advantage viruses have, especially those with RNA as genetic material, is they have an enzyme known as RNA polymerase. Basically, in common terms, this is a photocopying machine. Unfortunately, this enzyme is a lousy enzyme. So whenever it copies, it makes errors. Now, this is an advantage for the virus because when it makes errors in copying its genetic material, it results in mutation. Mutation is basically a change in the code that translates into proteins. If you take DNA viruses, on the other hand, they have the ability to cause latency. Latency means lying low. And how do they do that? They integrate into the host genetic material. And whenever the time is opportune, when there is stress or immunosuppression, they go into translation and fresh viral particles are produced. Basically, many viruses of the herpes family produce latent infections. In common terms, it's lifelong infection. And lastly, viruses have a wide diversity and ability to jump species, from animals to birds to mammals. So I think these are the 4 major advantages viruses are here to stay. Where do they exist? If you read the Dancing Matrix, Martins Hennig argues that they live in their own niches and corridors, and which is very true. If you go to South America and the forests of either Brazil or Colombia, there are hundreds of viruses which are awaiting to spread to the rest of the world. So they have their own niches. Over 30 years, I've realized that I have become an advocate for viruses. I will argue my case throughout this talk and tell you why it is so. They live in their own corridors and their niches, and it is human beings, because of their behavior, crisscross these corridors and invite trouble, and they go into niches where we invite them to cause infections in our tribe. They are abundant in nature and they are found in almost every living system that has been described, right from bacteria to fungi to unicellular organisms and amphibians, birds, mammals, you name it, they all have viruses. You will be surprised to know that 60% of the ocean mass is viruses. If you remove them, it will have a huge impact on diversity, and maybe we will not exist. So viruses are by and large beneficial unless we attract them to our uh, anatomy and make them cause disease. They are present on all the flora and fauna of the planet. Why do they emerge? Because we are in close proximity and contact with several other living species in the planet. So the argument people have posed is this results in a spillover. I ask the question, are they responsible for spillover? I will tell you that they are not responsible for spillover, because in this slide I'm talking to you about the primary causes of emergence and re-emergence of viruses causing human infections. They are viruses like RNA viruses, which result in mutations that gives them an advantage in terms of virulence. Primarily, it's human behavior, be it political, social, or economic. This is what results in viruses emerging from their niches. Several environmental factors, like constructing dams without having any public health concept on what it is going to impact in terms of vectors or in terms of changing the ecology, changing agricultural practices. We have elegant examples within the country of trying to increase rice cultivation. With that, we invite mosquitoes of a particular species, then birds migrate because there is stagnant water, and then it results in JE outbreaks. Global warming has contributed to increase in viral diseases, especially dengue. Deforestation, people going to forests and attracting diseases like Kyasanur forest disease, which was restricted to one taluk. Now it's found in 6 states in this country. So there are a number of other factors such as healthcare advances. We are using a number of devices. We are using immunosuppressant drugs. We do transplantations. All these result in infections, viral infections. Our greed for food production, especially in the mid-90s, when I lived the scare of mad cow disease in UK, we fed cows with offal, a non-vegetarian diet, which is processed remnants of the spinal cord and the vertebral column of sheep, which had scrapie, and this caused the prion disease, and that transmitted to human beings through beef eating. HIV is an elegant example of how human behavior results in transmission. The viruses on their own cannot jump from one person to another. It's human behavior that is important. And of course, the poor infrastructure in public health and microbial adaptation and change has also resulted in emergence of viruses. I entered the medical college in 1973, so I have made a list of the number of new viral infections or re-emerging viral infections in the last 47 years. 22 have emerged, starting from hepatitis B in 1973 to SARS-CoV-2 in 2019. Look at this. One virus is emerging or re-emerging every 2 years, primarily due to human behaviors. How do you tackle eternal viral threats? The 4 fundamental pillars on which we have to rest our case for tackling eternal viral threats are surveillance, efficient diagnostics, building laboratory networks, and understanding disease biology. Now, each of these points I'm going to discuss, and I'm going to lace them with personal experience of the advantage I had working for the last 3.5 decades. If you take surveillance as an example, the first lesson in surveillance in a massive scale that I learned was during the H1N1 pandemic. We were the only laboratory in Karnataka. Thousands of cases were occurring, and we were under a lot of pressure, receiving hundreds and thousands of samples. What all I learned in 30 years, I could not put to use because this was a huge, huge outbreak. Indeed, we processed over 100,000 samples in 2 years. But it was an opportunity to feed data for policy change. When we started, oseltamivir or Tamiflu was a drug that was scheduled and given only if a person was positive. Within a matter of few days, we had to put our foot down and say that prescription of a drug cannot be tagged to a laboratory report. Rest of the world was not doing it. And I remember Government of India changing the policy. It was a first time lesson for me to handle media and to handle administrators. It requires an entirely set of new skills. And of course, trying to coordinate with about 92 hospitals in Bangalore was a nice experience. But the surveillance experience I cherish most is what I've done in the last 5 years between 2014 and 2020 on encephalitis, which is a neglected disease. Julie Jacobson of the Gates Foundation calls it as the king of orphan diseases. India has had huge outbreaks of encephalitis since the early 70s, and we introduced vaccination for Japanese encephalitis in 2005. Following this, the contribution of Japanese encephalitis virus to acute encephalitis syndrome remarkably decreased from 35-40% to about 10 to 12%. But the number of encephalitis cases had not come down in the country. In fact, they were increasing, as you can see the blue line in the graph on the right. So it obviously raised a question on what is the cost for the remaining 80%. So we set about in a CDC funded project to ask this question. And what did we do? We first engaged with stakeholders, multiple stakeholders, state health authorities, central agencies, medical colleges, and district hospitals. Encephalitis is predominantly a rural disease, and patients get admitted to district hospitals. With the help of the government, we identified sites where we had to build a network. With clinicians and others, we finalized an algorithm and a testing strategy. We built capacity in all the district labs for basic tests, and we established a referral lab in every state capital because the states did not have the capacity to test for agents other than Japanese encephalitis. We established a robust sample transport system because patients are in rural areas, and then the testing facilities are in central areas. And of course, we streamlined data collection. We worked in 3 major high burden states of Uttar Pradesh, Assam, and West Bengal. We worked in close to 18 sites. Each state had a medical college or a School of Tropical Medicine in West Bengal as the nodal center. We built capacity in these labs for doing non-JE tests. The algorithm prior to our intervention was focused primarily on testing for Japanese encephalitis. And so we asked clinicians what do they want. They gave a laundry list. We pruned that down, and we added other known causes of encephalitis to the list, like bacterial meningitis, herpes, enterovirus, dengue, West Nile, chikungunya, and leptospira. So the output of our project, it showed that instead of having only JE and unknown etiology, we had totally at the end of this algorithm, 11 different outputs. We could identify the etiology. This is a snapshot of the work we did in 5 years, 14,969 patients who we enrolled, collected serum and CSF. We had 2 broad approaches, immunological approaches detecting antibody and antigen, in the serum samples because it's not possible to get CSF samples in everybody. But with our intervention, we were able to push CSF collection to 75%. And in the spinal fluid, antibody tests have very minimal specificity and sensitivity. So we relied on the nucleic acid based tests such as PCR. And what did we find? We found that we were able to get, using our algorithm, a total of 48% of cases being detected. I would like you to recall that prior to our intervention, only JE was being detected. The yield in diagnosis was only 10 to 12%. So overall, the application of the algorithm resulted in a huge increase by fourfold, 3 to 4-fold in etiology. And more importantly, Japanese encephalitis, scrub typhus, and dengue were the commonest pathogens that accounted for 73% of all etiologies. We had several outcomes in this project, very gratifying outcomes. The first was enhancing etiology from 60 to 48%, a threefold increase. Identification of treatable causes, especially in Uttar Pradesh, West Bengal, and Assam, mainly in Uttar Pradesh. 22% was scrub typhus. That's good news because it's eminently treatable with antibiotics such as doxycycline and azithromycin. This led to a huge policy change of implementing empirical therapy for scrub typhus in UP, and the government of UP issued circulars, which is in the center you can see in Hindi, that after a patient is admitted and samples are collected, they should be administered doxycycline or azithromycin, as the case may be. It also resulted in a major policy shift of the government of India, the Vector Control Program and ICMR, in telling that the algorithm that we have used is very appropriate for encephalitis. Therefore, it should be implemented across the country, and that was very gratifying. We built capacity in 20 sites and 4 Apex laboratories. So this work is continuing now. We handed over it to the state governments and they are supporting the program. We also took the advantage of availability of technology to develop a digital platform which can be used on a mobile phone or a computer for collecting data and rapidly reporting results to the clinicians. So that's the surveillance experience, kind of experience we had. Moving on to diagnostics. As Krish mentioned, we have one of our areas of work has been in developing indigenous kits. The first commercial Japanese encephalitis kit was developed in our laboratory. We reduced the time from an in-house test from 36 hours to 3 to 4 hours. We made the reagents robust. We had very good sensitivity. The WHO used our kit in entire Southeast Asia. Indeed, this was the forerunner of the NIV kit. And we also shared our technology with NIV, and now National Institute of Virology is manufacturing these kits. Encephalitis diagnosis, as Krish said, has been a challenge. Multiple agents, and clinicians always want diagnosis on a single specimen. So we embarked on a very ambitious project to develop a chip that could simultaneously detect 16 organisms. It was one of the first when we did that about 14 years back. Now, of course, a number of commercial technologies are available. Basically, we designed primers and targets for PCR from these bugs, from their virulence regions. We standardized a uniform cycling condition, and then we developed a hybridization platform. And based on that, it developed spots, brown spots, which you see in the inset there. This encephalitis chip was subjected to evaluation. It had a sensitivity of 98% and a specificity of 94%. It only missed 3 rabies cases, but most importantly, in the second group in the table on the right, where there were suspected cases of encephalitis, the existing test drew a blank. This chip was able to provide diagnosis up to 63%. So this is now with the company as a research service and a diagnostic service. One of the other major reasons of our ability to manage outbreaks in the last 2 decades has been building global networks, laboratory networks. Now, the best example is the network for polio labs, which has a great role to play in eradication. There are over several hundreds of labs across the globe. They are still functional. They're still continuing to look for polio viruses in clinical patients. I had the opportunity of establishing a WHO CRO network, the first of its kind for Japanese encephalitis, in collaboration with WHO, especially under the guidance of Dr. Nalini Ramamurthy. Since networks are very important, the country also has now many networks. You have seen the impact of the ICMR Virus Diagnostic Research Laboratory Network in the COVID diagnosis. There's a network for arboviruses. There's an integrated disease surveillance program network and the HIV networks. And we have been part of all these networks and contributed our might to the growth. Since it's a memorial lecture on Dr. Suniti Solomon, I must say that I drew a lot of inspiration from Dr. Suniti Solomon, and our lab now performs a number of activities under the National AIDS Control Program. Apart from being an integrated counseling and testing center, which we started in 1986 and became a NACO center in 92, we do a number of activities. We provide quality assurance for HIV testing. We do quality checks for samples periodically from labs that are doing testing. We confirm the status of indeterminate samples by Western blot. We do a large number of trainings for laboratory technicians and counselors. We are a repository and we have samples HIV, and we have been involved in HIV sentinel surveillance. And one of the other major activities we have undertaken in the last decade or so is to serve as a member of the consortium that NACO established for kit quality testing. We do quality tests on every batch of HIV, hepatitis B, and HCV kits that are procured by the country using a large panel of samples and a standardized protocol, which is an accredited laboratory. The next thing that if we have to tackle pandemics and threats is to understand disease biology. I am taking 2 examples, our personal examples. H1N1, during the outbreak and after the outbreak, we asked a very important question: why is pandemic flu different from the regular flu? Because mortalities were higher. So we hypothesized there must be viral factors, host factors, and host genetic factors. In this, we address the issue of viral factors and host immune response. For a period of 4 years, we systematically isolated virus and subjected to sequencing. And we looked at sequences between mild, moderate, and severe patients. And lo and behold, we did find that the mutations were distinct based on the category the patient belonged to. And we identified a very unique mutation, the D239G, which was present in the hemagglutinin protein of the virus. We also looked at host response between traditional seasonal flu and the pandemic flu. And these are the clinical characteristics. We chose the groups in such a way that their characteristics were similar. And to summarize the findings, we found that those with severe pandemic influenza, H1N1 or the swine flu, they mounted a very robust, aggressive, pro-inflammatory response, especially resulting in flooding of the lungs and the patient going into ARDS. The seasonal flu, on the other hand, did not exhibit such a robust response. And this experience came in handy during COVID time. So that brings me to COVID and the coronaviruses. They've been around for a long time. most of them causing mild infections for 4 decades, common flu and diarrheas. Turn of the century saw emergence of SARS, which spread to many countries, and fortunately we contained it. And then in 2012, we had the Middle East respiratory syndrome. And you all know that these viruses are widely distributed and they are present in humans and animals, and spillover events occur from animals and birds to human beings, like it happened in the wet market in Wuhan, a crowded area where large number of animals, birds, and humans were present. How does the disease spread? I have learned a very valuable lesson. I thought I should share it with you. We looked at 3,400 cases of COVID in Karnataka prior to lockdown. Why prior to lockdown? It was a controlled environment. And in this environment, we found that 90% of the people were asymptomatic and 10% were symptomatic, like the rest of the globe. The asymptomatic people were younger, the symptomatic people were elder and had comorbidities. And the main spread, when we did an interesting analysis known as cluster analysis, we found that symptomatic patients are the main people who are sustaining the outbreak. What do I mean by sustaining? Both asymptomatics and symptomatics transmit. Let me explain this graph for you. This is a cluster diagram. A cluster is a collection of cases with a single source in the center. A red dot in the center means the patient is symptomatic. A yellow dot means asymptomatic. In the 46 clusters where we had contacts and analyzed in detail, majority of the clusters had a symptomatic patient in the center. Not only that, the clusters were bigger when the patient was symptomatic. And also, very interestingly, symptomatic patients in the center of a cluster contributed to 82% transmission, whereas asymptomatic people contributed only to 17% transmission. Moreover, in the people who get COVID, if they got it from a symptomatic, many of them turn symptomatic themselves. On the other hand, if I got it from an asymptomatic, our analysis very unequivocally said that you are more likely to be asymptomatic. Look at the last column. The average dispersion or the probability of transmission from a symptomatic patient to a non-immune person is 14.3. It is 4-fold less for asymptomatics. They also transmit, but to a lesser extent. I think it's a very valuable lesson that tells you that we should be chasing, hunting, testing symptomatics and isolating them. Asymptomatics are also important, but if we have to curtail transmission, we have to be after the symptomatics first. COVID has taught us a lot about how a virus produces disease. Let me act as an advocate for viruses. Most often, in most viral infections, viruses do not multiply and kill human cells. That's definitely there, but the proportion varies. In the case of COVID, there are 4 mechanisms, as this diagram shows you. Direct toxic effect of the virus on cells is very less. Virus also alters the homeostasis, especially in the blood pressure circuitry, because it binds to blood pressure receptors. It damages human blood vessels, resulting in clot formation. But above all, the most important factor is that it has a profound effect in some individuals on the immune response or the defense response, resulting in a very aggressive response that causes most of the problems, flooding of the lungs and multi-organ failure. In fact, it's very interesting that this is a concept that was taught and vocalized by the father of modern medical microbiology, Louis Pasteur, who was actually a brewer, a fermentation specialist. He did a lot of work on modern microbiology. We owe a lot to him. On his deathbed, he said, "The germ is nothing, the terrain is everything." Look at the spectrum of disease caused by COVID. 80 to 90% asymptomatic. Lucky people, they mounted the right response. 5 to 10% of the people, moderate clinical picture. They mounted a slightly inappropriate response. The unlucky ones are the critical people, the 5%, who mounted a very aggressive immune response and landed up with problems. That brings us to the kind of challenges we faced with viruses being as the eternal reality. I see there are at least 4 major areas of challenges COVID has thrown up. Epidemiological, I think the biggest challenge has been in quickly tracking and tracing people, let alone testing being a huge challenge because it takes time and is resource intensive. In terms of clinical, it caught us on the wrong foot, especially low middle income countries like India. We are a country where advanced healthcare is available only in major cities. 40% of our districts did not have intensive care units. So we did not have a system to effectively manage beds when there's a huge surge of cases. It's a pity that we do not have piped oxygen supply in most district hospitals. And of course, we don't have an effective antiviral drug. Social challenges like HIV, it has posed several social challenges, much akin to HIV, reactions of panic, stigmatization, discrimination, denial, anger, depression, mental health issues in COVID have been so huge that it has brought mental health back into focus. And of course, the biggest of all challenges have been the inequities in healthcare availability across the globe. Challenges in prevention, new behaviors are always a challenge. We have never used to distancing, social distancing, staying 6 feet away from people. We have never had the habit of wearing masks. Even today, mask is worn as a decorative ornament around the neck. It's not worn properly. And of course, we do not have a vaccine. And we have to also remember that in the context of prevention, protecting the vulnerable. Who are the vulnerable? The elderly with comorbidities. I think this brings a huge challenge on the youngsters to behave responsibly, not to carry infection home. Rather than project a negative picture, I thought I will end with some positive notes. Every calamity, there is an opportunity. And look at COVID, the opportunities it has thrown up. It has brought public health to the center stage. It has used technology very effectively for tracking, tracing, and quarantining people. It has brought a number of experts to the table and make them examine data and drive interventions on data, based on data, something which I personally enjoy. Communication, workshops, honest, regular dissemination is very important. We are realizing that. Unfortunately, most of our messaging is based around fear and panic. I think it's an opportunity to do positive messaging. And of course, reiterate the necessity for syndrome surveillance. In terms of clinical opportunity, we have learned a new method of consulting patients, telemedicine. We are adopting uniform guidelines to reduce mortality. We have learned to rotate our workforce, especially in the hospitals, in the clinical setting. Half work for a week and the other half take off. And of course, it's a huge opportunity to upgrade our healthcare facilities. Here is an opportunity to integrate alternative systems of medicine, which we have been practicing for centuries. But we have to bring rigor of modern medicine to evaluate the evidence base of these systems of medicine. A huge opportunity in testing. Around the time I'm retiring, it's a personal satisfaction that the government of India has now made it mandatory that every medical school should have a virology laboratory. Madam Suniti Solomon will be very happy in heaven to hear this. We know how to use tests now. We have to use appropriately. We have to use minimum testing and maximum intelligence to get the best outputs. In terms of social opportunities in society, it's a huge opportunity to policymakers and politicians to increase allocation to health. Come on, this is biological war. We spend 40% of budget on regular defense to protect ourselves from wars and to tackle wars. Now, here is a biological war from organisms that are invisible. I think if there is one less takeaway message for everybody, it is to ensure that we increase allocation for health. We should learn to respect environment in the planet where we live, other life forms, the flora around us, not to deforest. The more we foray into forests, the more we indiscriminately construct, the more we indiscriminately use animals, we are at risk of getting infections. Stress has been one of the major social challenges. We are now believing and we are telling the world that yoga is a stress buster. Universal coverage is health coverage is a social opportunity. The biggest opportunity is to develop appropriate behavior change, communication, and to provide mental health services to everybody. Opportunities of prevention in the context of COVID, avoid the 3 C's, closed spaces, crowds and close contacts of the highest degree of transmission. How can you beat it? Watch your distance, wear a mask properly, change your mask at the right time, wash your hands frequently. And of course, we'll have to hunt down symptomatics and get them tested and isolate them. And hopefully we'll have vaccine one day. Before I end, 2 minutes I'll spend on the kind of initiatives that have been taken. I must mention the name of Nathan Wolf. He's at Stanford. He's in California. He has started a firm known as Global Viral Forecasting, which uses engineers, big data managers, epidemiologists, and what he's experimented a decade back is to set up listening posts across the globe where data is mined from social media, from Google and other places to know if there is an outbreak. Somebody may tweet or message saying, I'm unwell, I have a cold, I'm not coming. Then if you use this data and use geo-positioning, you will realize that in a small area, in a town, there are 40, 50 patients with the disease. You can swoop down, use technology, diagnosis, and to arrest outbreaks. I think this is fantastic technology. We should look forward to this kind. It provides opportunities for forecasting outbreaks and pandemics. To summarize, viruses are an eternal reality. They are here to stay. We have to learn to live with them and live around them. And how do we do that? Human activities are the most potent driving factors of viral disease emergence. There are innumerable examples of it. Social, economic, political, climate, and technological and environmental factors all shape disease patterns and influence emergence. We should learn to harness technology, work together, cutting across disciplines for diagnosis, early detection, and prevention. Understanding and responding to disease emergence requires a global perspective, as COVID has shown, conceptually and geographically. And in summary, the current global situation, unless we change the way we behave, favors disease emergence. Before I end, I would like to acknowledge all my teachers, right from Professor Suniti Solomon to my teachers in JIPMAR, my family, my little sister, who gave me a purpose in life and motivated me to take up virology as a career. She died at the tender age of 14 with fulminant hepatitis B infection diagnosed incidentally by Professor Solomon and Professor Tyagarajan. My parents, who were enormous support and implemented values, my siblings, especially my brother, elder brother Kumar Swami. Many of you may know him. He was my personal mentor, and my wife, Prasuna, and my two sons, Ashay and Abhinay, who allowed me enormous freedom to spend more time in the lab and less time with them and encourage me all throughout. All my collaborators at NIMHANS, Professor Chandramukhi, Professor Shankar, Professor Gauri Devi, Professor Satish Chandra, and my colleague, Dr. Anita Desai, requires special mention. She has been with me for 30 years and has continuously worked hard along with me, and I've enjoyed her inputs into the various aspects of work that I did. My other colleagues, late Dr. Madhusudan, Dr. Rita and Manjunath, all my students, my international collaborators, Dr. Barbara Johnson from CDC, Dr. Nalini from WHO, Dr. Tom Solomon, and the huge team of investigators led by the CDC India group, Dr. Padmini, Dr. Kela, and all the states where we could do the encephalitis work, a total of 60 of us. My staff have been exceptionally committed and supported me, and above all, the great institution at NIMHANS, which allowed me to blossom and express myself in a way that I have enjoyed my 37 years of association. Thank you for your attention. Stay safe and stay healthy.
Thank you very, very much, sir. I think we are overwhelmed. This has been such a great evening. I have been receiving continuous feedback from people from all walks of life who have uh, rendered such great feedback, saying a layperson could understand what you spoke and you're such a great teacher. We are so thankful that you were here this evening and you were able to give us this message, sir. We are grateful. I would now request Dr. Krishnamurthy to take in questions for you, sir, and moderate the following session for about 10 to 12 minutes. Dr. Krishnamurthy.
Yeah, thank you, Dr. Setu. Lakshmi, Professor Ravi, I think that was such a wonderful lecture, so lucid, so easily understandable, and so inspiring. Thank you so much. The first question I have is from Dr. Janaki Pillai. Do you believe that given our experience with COVID and the challenges you outlined, there will be greater investment in public health? I know you called for this yourself.
I am a perpetual optimist, Krish. I believe governments will wake up. This is a wake-up call of the century. Last century, we do not know because there are very, very hardly any or few survivors of the Spanish flu. I hope people will realize that in the interest of global economy, growth, and health, investing in public health and investing in health in general is probably going to be a very important initiative that all governments should take.
Thank you, and I hope some of the policymakers are listening. There's so much talk about a vaccine, Professor Ravi, and of course, many people are eagerly waiting for the vaccine with regard to COVID-19. How important is a vaccine in the scheme of things?
A vaccine is very important. Let me tell you, Krish, that human beings are an extraordinary selfish species. They do anything to survive. And I think that's a testimony to their survival for centuries. Having said that, the most attractive and the easy proposition for prevention is a vaccine. It's very, I don't have to tell you, being a psychiatrist, that changing behavior is far simpler as a strategy, but very difficult to implement. So people look always for a quick shot to develop protection by producing antibodies. So that has been the tradition, and that has been the rule for the last century for a variety of viral infections. Coming to COVID vaccine, we believe that antibodies are protective. I hope that belief will be borne out at the end of December, when phase 3 trials will tell us how many people developed antibodies, how effectively are antibodies produced, to what levels, and are they protective. this will be answered in the phase 3 trials of the 6 vaccines that are going on currently. What we do not know, the black box, is what is the role of T-cell immunity. How important is it? Evidence is emerging that cell-mediated immunity and T-cell immunity is also very important, although it's very, very difficult to measure it on a large scale in every individual. So antibodies remain a surrogate. I am hoping that the vaccine will at least show certain amount of protection. That's the first challenge. The second challenge is scaling up production, which is going to take 2 to 3 months. And then, of course, the big question, who do you give it to? Are we going to prioritize? Is it going to be available for every nation? These are daunting challenges, but I'm sure we'll find a way out. But in my opinion, nothing before April 2021. Over.
Right, sir. Thank you very much for that lucid answer. There's a question from Vasudevan in YRG, that people are becoming very careless, and is risk communication needed, and is it important?
Absolutely. Vasudevan hit the nail. Risk communication is very important, but I must tell you, I mean, I should not talk in front of Krish. Risk communication, it should be in a way that it is not threatening. At the same time, it is enabling in terms of changing behavior. Dr. Suniti Solomon and all the people who have worked in HIV will tell you this, that behavior change is not easy. Knowledge does not translate into practice. In between, you have to cross a bridge known as attitude. So your knowledge should change your attitude, only then your behavior will change. And I think this is something that we have neglected in the country. Our messaging has been our biggest weak link. Only fear-based messages. Statistics. You open a TV, you'll see thousands dead, 1,000 cases detected. Nobody says, look, even if you get COVID, most likely you will recover. You don't have to get COVID if you do A, B, C. That kind of messages. And we have not harnessed on great champions to pass this message across. Over.
Dr. Bhagyashree wants to know about IgG antibody after COVID, and how soon does one develop it, and so on.
COVID is a virus that threw viral immunology to the wind. It gave us so many surprises. For most RNA viruses, acute viral infections like dengue, Japanese encephalitis, chikungunya, usually antibodies start appearing second or third day. They reach a peak by second week, and then they are sustained for at least 6 months to a year. COVID, for some reason, is very delayed. Antibody response is very delayed. The earliest antibodies, minimal amounts appear at the end of a week or 10 days. They start gaining momentum by second week, and they probably reach optimal levels by third week. And very surprisingly, 10% of people do not develop antibodies. We don't know why. And more people with asymptomatic infections, whether they cleared the infection using an innate response is a big question. Their adaptive immune system was not educated, so they did not develop a good and robust antibody. These are the people who are now coming back with reinfection. IgM, for some reason, does not appear early. It appears along with IgG. So there's no advantage of detecting IgM in COVID. IgG almost is the first antibody to appear, and it takes at least 2 weeks. And we don't know how long it will last. We have experienced only a few months. We know that in some of the studies, antibodies have started waning after a matter of 4 to 8 weeks. Now, this is a cause for concern because we have to hit the right immunogen for a vaccine. Over.
Thank you, sir. There's a question on whether we can achieve herd immunity before we get a vaccine.
It looks like it may happen. Zero surveys in many parts of India in March, April, May said 1 to 2%. Repeated 3 months later are saving 20 to 30%. And in another 3 months, hopefully we'll hit the magic mark of 50 to 60%. But now people are revising their calculations and they're telling you may not require 60% of people. If 40 to 50% get infected, then the curve will start flattening, but it will linger on. It will smolder like the H1N1. It is not going to go away. Let us be very, very clear about it. Oh.
Does the severity, this is from myself, does the severity of the clinical symptoms change as more and more people get infected in the community?
I'll flip the question around. You're asking a question, is the virus mutating and adapting and becoming more virulent? The answer is no. There is no concrete evidence. We have sequenced 100 genomes across the globe. Thousands of genomes have been sequenced. People have been looking very hard at sequences from mild, moderate, and severe. They have not found anything worthwhile. Remember, this is not flu virus. Flu virus is a highly mutating virus because it has 8 segments. There is recombinations and mutations. So that's a very deadly combination. Here, it's a single standard genome, comparatively stable compared to flu or HIV. Not many mistakes are made by the polymerase. So as Louis Pasteur said, it is not the seed, it is the soil. It is you and I who decide which compartment we go to. Over.
Thank you. Very eloquently put. Many people are asking in the chat box about the various conspiracy theories about the virus, whether it came out of a lab. You want to take that question?
The honest answer is nobody knows. But let me argue scientifically. Normally, when people tinker with viruses, they leave behind some telltale evidence, a little marker, so that they can track it in the community. Very close examination has shown that there is no such marker. In fact, Science and Nature brought out articles saying that it's not likely to be a tinkered virus. Let us look at it the other way around. This virus has 98.5%, 98% homology to bat virus. Bats were there in the Wuhan market. It has 99% homology to the pangolin virus. It is believed the virus jumped from a bat to a pangolin, which is an anteater family animal, and they were also sold in the Wuhan market. So we have more evidences to say that sequence homologies are closer to animal viruses rather than to a genetically engineered virus. Over.
Thank you, sir. Question from Mr. Jawahar asking about the skepticism in the US about the vaccine. Do you have any comments on why there may be skepticism around in certain parts of the world about the vaccine?
This skepticism is because we have not understood the immune response completely. We really do not know what is a marker of protective immunity. I fondly hope antibody is not a red herring, because that's the one we are chasing. That's the one we are measuring. I think that there is some basis in the skepticism. Having said that, let us wait for the results of the phase 3 trials, which will tell us how good the response is and how wide the response is in terms of population. Over.
Someone in the chat box has asked about vaccines for children. Do you have a view, sir?
I am a proponent of vaccination because it protects against at least a dozen serious illnesses, whether it is polio, measles, mumps, rubella, or Japanese encephalitis, or hepatitis B, or for that matter, certain, selective vaccines like chickenpox vaccine, they all reduce morbidity, they all reduce mortality. So I think that is the low-hanging fruit that is available for protection. And I think one should grab that fruit. Oh.
So if there was one health behavior communication message that you would want to give everyone, what would that be?
I am very fond of this message, which I have always said right from my HIV days. Our protection is our responsibility. It's a non-judgmental statement. At the same time, putting a whole lot of emphasis saying that what you do will determine whether you get infected or not. And it's now, I translated into Kannada, it's being used in Karnataka. Nanna Surakshita, Nanna Javabdari. My protection is my responsibility. And I think that's a huge statement because we need to protect elders in the house. We know that they are the most vulnerable. So the way I behave, the way I conduct myself will determine whether I get infected and more importantly, whether others in my house get infected. Over.
Thank you, sir. I think that's so important that we protect each other. There's a question from Dr. Valarmati. I'm sorry, I'm just losing that for a moment.
Sorry, Dr. Krishnamurthy.
Yes.
No, if after Valarmati, may I request Mr. Kannan also would like to ask a question after Valarmati's?
Certainly. So I'll just finish Ms. Valarmati's question. On your thoughts on eradication of diseases as we talk about viruses being eternal.
It is, you see, eradication is possible when the virus is a single type, it does not mutate, and there are no reservoirs. Classic example, smallpox. I'm so fortunate in my lifetime, I've seen two diseases eradicated. One is smallpox, the other is polio. Polio was a humongous effort in the global community. Everybody participated, and only through sheer vaccination we got rid of polio. Are we going to eradicate other diseases? Whatever I went through, the list of my experience, I'm very, very skeptical because human behavior has to change. And that's a huge challenge. Will we stop going into forests? Will we stop ravaging the environment? Will we stop crowding with animals? I don't know. I don't know whether eradication is a possibility. The optimist that I am, I hope human behavior will one day change and we will get rid of diseases.
Thank you, sir. Mr. Kannan?
Good evening, doctor. Could you tell something about the protein layer on the virus? Can it be a source for a solution? Because rest of it is only a RNA matter, but the protein layer, what can you do with it?
Thank you, sir. I did not go into technical details. As I said, virus is a piece of genetic material wrapped in a protein coat. And in the case of coronavirus, it additionally has an envelope. I call it a gentleman virus. It wears a coat. On the coat, it has studded important protein known as spike protein. It is a globular protein with a stem. The stem is embedded in the envelope and the head projects out. This is the most important protein because virus attaches to human cells and animal cells using this protein. We call it the ligand or the key to open a lock. All protection is invested in this protein. This is the most commonly used protein for vaccine production now, either in terms of producing the protein in a mass using biotechnology approaches, or expressing this protein on another virus, a chimeric virus, and then injecting it so that that virus does not produce disease. When it multiplies, COVID protein is exposed and you develop immunity. So there are many, many approaches being taken. But you're right, the surface protein of viruses are most often the targets for immune response because the antibodies will neutralize their entry. Over.
Very interesting question on whether the rapid trials for the COVID vaccine are compromising safety.
The honest answer is, we do not know. Let's wait for the trials to be over. But you must have seen in the press that temporarily they were halted whenever an adverse event was reported, investigated quickly, and then cleared. So a conventional trial normally takes a year and a half. We are compressing it by 1/6 to 3 months or 6 months. So how much of compromise will be there in the quality of the vaccine, in safety, only time will tell. Having said that, science has advanced so much that we have learned to compress many things, right? From zipping a folder on the computer. I think this is the first time we are experimenting. Let us hope we succeed. Over.
I was just reflecting, and there's a question about this as well, about man's proximity to animals in two ways. One, of course, is living with animals as pets, and that's the question that has been asked. But the other, of course, is the industrialization of animal use for food and how that is contributing to virus infections. You mentioned CJD as well. Could you please comment on both these things?
As regards pets and the threat of getting infected or infecting pets, it is very, very minimal because most of the people, at least most, not all, most of the people who keep pets make sure they take care of the health of the pet, they get it immunized, and they themselves are also immunized. So the spillover from human to animals, there is no clear-cut evidence to that as of now. Okay, by natural process of cohabiting with animals. Okay? The reverse is very dangerous, especially animal being used for industrial purposes of food production, crowding, and it's almost, you can predict the hotspots in the world where the next virus will emerge. And unfortunately, Southeast Asia is top on the list. The second would be Africa, people venturing into jungles. The third, the most dangerous in my opinion, would be the South American jungle. God forbid we don't go and try to colonize these jungles. We are in for trouble because there are hundreds of viruses there which people do not even know what they are. So I think if we respect our boundaries and respect life of animals and crowding we avoid, we will be able to avoid future pandemic threats. Over.
Sir, you very kindly referred to yoga as a way of stress management, and I think that's today a global message. You also mentioned Ayurveda as preventive health, and there's a question in the chat box about steam inhalation as a preventive, technique. Your comments on that?
Steam inhalation as a preventive strategy, there is no objective evidence. It does good for the respiratory system. It opens all the channels. It liquefies thick secretions, which are important as an innate defense mechanism to have liquid mucus rather than solid mucus. To that extent, it definitely helps. To believe that the temperature of the steam will kill the virus, I think is stretching it a bit too much, in my opinion, from scientific evidence. Over.
Thank you, sir. And I suppose one of the things that we have to all keep in mind is also that those people who inhale steam are likely to be more careful in many other ways as well. So they're probably better protected.
Very true.
confounders, as we say in research.
Yes.
I'll take a final question, sir, about plants and viruses. Do plants have a role at all?
Very nice question. We have no objective evidence on record that plant viruses infect human cells. Plants have plenty of viruses. Every vegetable has a virus, and many of these plant viruses incidentally have been discovered by Indian plant virologists, whereas the tomato virus or the bindi virus or the potato virus or the brinjal virus, I mean, there are innumerable discoveries on plant viruses in India. In fact, history of virology started with the tobacco plant. Vijaynik first found some mosaic pattern on tobacco leaves, and he asked the question, what is causing this? And then he actually extracted the fluid, looked in the microscope, didn't find anything, and he called it Contagium vividum fluidum. And he said there is a soluble factor that is transmitted from one plant to another. But there is no record. There is no record of plant viruses causing infections in humans. That threat is not there. Over.
Thank you, sir. And I have one final question because it emerges from an elderly patient of mine, about 87 years old. And when we were talking about this, he said, you know, doctor, if it's in my karma to get this and this is what is going to take me away, then so be it. Is there such a thing, do you believe, for virus infections as inherent protective factors within humans, which determine how we respond to the infection?
Very nice question. If you look at human physiology, immune system in the first decade takes time to evolve. It is learning to wage the war and fight the battle. So they are very vulnerable. As we age, we call it senescence. Beyond 50, 60, many systems in the body age, and one of them first to age is the immune system. If you can train the immune system to go on staying at a particular threshold, you can train it. How do you train it? By exercise, good food, minimizing stress, and one of the most important things required for immunity, of course, is good quality sleep. Nobody talks about it. And supplementation. As we grow old, we don't absorb vitamin B12, a crucial element for immune system. Our vitamin D levels are abysmally low. We need to supplement that. Vitamin D is a great modulator of protective immune response. There is ample evidence. There are actually meta-analysis saying that vitamin D is a very, very important dietary supplement when it comes to appropriate immune response. Exercise, yoga, good food habits, avoiding substance abuse, all these are factors that will help us to maintain our immune system at optimal performance. Over.
Thank you, sir. And which also brings me to think about how our lifestyle in the past used to include leaving our footwear outside the house, bathing as soon as we came back from outside, and closing our mouths when we spoke to someone, especially the elder. So I think our ancients knew some of these lessons much before we invented social distancing. Thank you so much for not just a very lucid and wonderfully presented lecture, but also for taking all these questions so patiently and answering them so well. It has been an enormous privilege. Thank you, sir.
So much.
Thank you to Setu Foundation.
And honor to interact with all of you and to remember Madam on this occasion.
Thank you, Professor
Dr.
Ravi.
Setu Lakshmi.
Thank you, Professor Ravi. Thank you, Dr. Krishnamurthy. That was such an engaging discussion, steering all the conversations with questions and answers to best suit our people who've actually filled our chat boxes with questions. Professor Ravi, you have actually connected one last dot for the evening for us. Setu Foundation's theme is unity of life. We believe in respecting all lives and boundaries. And I think you helped us to understand that further in your talk. I would like to request Mr. Kannan to tell us how this theme came about, the title, Virus, the Eternal Reality. Sir?
Madam, one request, one suggestion from my side. If I have not answered any questions, if you can transmit by email, I will try and answer them for the people.
Will do, sir. We'll bear that in mind.
Thank
Anand,
you, sir.
sir.
Can you hear me?
Yes, sir.
This topic came about in another strange way. On 22nd August issue of The Economist, the title cover story was The Aliens Amongst Us. It was fully about viruses. And one small sentence really it was so elegant. I'll just read it out. That viruses are best thought of as packages of genetic material that exploit another organism's metabolism in order to reproduce. They are parasites of the purest kind. They borrow everything from the host except the genetic code that makes them what they are. They strip down life itself to the bare essentials of information and its replication. The sentence is so beautifully elegant. They strip down life itself to the bare essentials of information and its replication. Abundance of viruses is anything to go by. That is a very successful strategy indeed. So this particular paragraph, it struck that viruses are everywhere and they're eternal. I think we will find them in many parts of the universe also. So this theme emerged out of this particular paragraph and this particular article. Thank you.
Thank you, Kannan, sir. I will now request Kamla to render the vote of thanks. A little introduction about Kamla. She's a teaching professional for the last 31 years, retired, but she continues her journey fervently by inspiring many lives that she comes across every day. She presently leads our Chetna program, which is a Setu Foundation initiative that works for children. Kamla?
Yes, I do. Thank you. Yes. Good evening to you all, distinguished ladies and gentlemen. It is a great honor to virtually be present amidst this august team of great achievers. I welcome Press Officer, the U.S. Consulate General Chennai, Madam Corey Beckel, respected Professor Dr. V. Ravi, NIMHANS, Bengaluru, Dr. E.S. Krishnamurthy, founder and CEO of Buddhi Clinic, Mr. S. Kannan IRS, Ms. Setu Lakshmi, MD, Setu Foundation. I take this as a very personal privilege and a great honor indeed to deliver the vote of thanks, and that I can be here in association with the first memorial lecture hosted by Setu Foundation for none other than late Dr. Suniti Solomon, who many of us saw as a person to admire, a role model who woke India up to the HIV threat. Let me begin with a big thank you to Professor Dr. Ravi. From day one, sir, you have consented to be our speaker, expressing your sincere interest to honor Dr. Solomon, who's been your teacher. It was indeed enlightening to hear and to know so much about these unknown facts that you have thrown light upon, these real bugs that have come to stay. And it also sends us the strong message that it is very important for all of us to behave ourselves in every sense of the word. I also would like to mention my deep appreciation to Ms. Corey Beckel for making her presence felt at the Dr. Suniti Memorial lecture today. Thank you, ma'am. Further, special thanks to Dr. E.S. Krishnamurthy, who introduced us to this distinguished speaker, Dr. V. Ravi, a more appropriate person we never would have found. We are grateful to your acceptance to be here today amidst all your work pressure and the promptness in introducing us to this great speaker and offering to guide us through this event. Thank you, Doctor. And we also are grateful to you, sir, for being here to propel and steer this program ahead for us. I wish to express my sincere thanks to Mr. Kannan for sowing the seed of having this memorial lecture for a great soul like Dr. Suniti Solomon. It was your thoughtful gesture that has brought all of us here together to appreciate and pay our respect in the most befitting manner to Dr. Suniti Solomon. It's been a great evening, just bringing the best minds together. It has added so much of special energy. The significance of today is that you have contributed to set a mark of respect to one of those doctors of India who has created history. Dr. Suniti Solomon, a remarkable woman who succeeded against all odds in making her voice heard throughout India. Sir, you made us realize how significant her role was in making the medicine board aware that India faced an HIV epidemic on an undreamt of scale. Medicines can cure diseases, but it needs a doctor to cure a patient. Dr. Solomon showed us this through her actions. I also wish to express my gratitude to Madam Sethu Lakshmi, MD Sethu Foundation, for providing this platform to honor Dr. Solomon. We have been fortunate enough to be backed by a team of very motivated and dedicated colleagues. Thank you one and all. We appreciate your contribution and we also acknowledge it. I, on behalf of Sethu Foundation and the entire supporting and sponsoring organization, the team, crew, and participants, and on behalf of Project Chetna, extend a very hearty vote of thanks to all speakers for joining us today and giving us your opinion and findings. You have made a difference. I end with this special final note: life is precious. Every life is important. You, Dr. Solomon, were the flag bearer of hope who never let us down. Can we ever forget you? Never. You will always be our guiding star as we walk this path of service to mankind. Thank you to one and all.
Last but not the least, we have successfully completed the first memorial lecture with no technical issues. So we are very grateful to the tech team led by Mr. Sridhar and by Mr. Prem. Thank you very much. And just to make the last announcement, the entire session today is recorded, will be available for viewing. We will also be circulating this because you'll have shared your email addresses with us while registering. It will also be available on YouTube as and when you would like to view it again. It has been a memorable day. Thank you one and all.